Bismillahirrahmanirrahim. 9700 November 23 paper 2-2, and this is the first video in which we shall discuss the first three questions. Question number 1. During interphase and mitosis of the cell cycle, the chromosomes within a cell go through a number of changes. Each chromosome is composed of DNA complex with proteins. In interphase, individual chromosomes are too diffuse, long and thin to be visible using a microscope. In this stage, the chromosome material is known as chromatin. State the term used to describe the proteins that are complex with DNA and form part of the chromatin, histones. So you just wrote histones, you got a mark, or you said histone proteins, you got a mark for that as well. When viewed through a microscope, a chromosome is most clearly visible during the metaphase stage of mitosis. Complete figure 1.1 to produce a labeled diagram of the metaphase stage of mitosis in an animal cell with 2 chromosomes. For 3 marks, this is the diagram that was expected out of you, and the labels that were expected, and the drawing that the 2 chromosomes, and then made up of 2 sister, one chromosome is made up of 2 sister chromatids. So there will be 4 chromatids, and then the spindle fiber and the centriole. Then it says outline the changes that occur to the structure and behavior of a chromosome from the start of S phase to the end of interphase during prophase of mitosis. So first of all, we had to give, this is 3 marks. Start of S phase, that means DNA replication will take place. To the end of interphase, that means G2 will have also taken place. And then prophase of mitosis will have also taken place. So we got to change, we're going to talk about the structure and behavior of a chromosome. Don't tell me about nuclear membrane and anything else. You're not supposed to be talking about that. So DNA replicates, got you 1 mark. New molecule of DNA with histone proteins and identical chromatids form. Then chromosomes or chromatids condense, become visible as 2 sister chromatids, become attached to the spindle fibers, and the spindle microtubules attached to the centromere. This is how we'll word it. DNA replicates, DNA associated with histones, identical chromatids form, chromosomes condense, becomes visible at 2 chromatids, and becomes attached to the spindle fibers. Question number 2. It says keratin is a structural protein in feathers of birds. Keratin polypeptides are composed of a high proportion of cysteine amino acids which have a sulfur containing R group. Keratin polypeptides form filaments. The 2 main types of keratin filaments are alpha keratin, which consist of many alpha helices, and beta keratin, which consist of many beta pleated sheets. Keratin can be classified as alpha and or beta keratin based on a study of protein structure. Suggest the level of protein structure used to classify a protein as alpha. So that would be the secondary structure, because alpha helix and beta pleated sheets are in part of the secondary structure. B part, protease enzymes hydrolyze proteins. Many proteases are able to hydrolyze more than one type of protein. Suggest why it is possible for a protease to act on different proteins. What makes sense? Said the active site may be flexible for binding to very similar substrates, or the proteins have a similar shape, so fit the active site. Or you could have said the active site and the substrates have complementary shapes. If any enzyme can work on a substrate, basically the reason is that the protein, the active site of the enzyme and the substrate have complementary shapes. Or you can say enzyme complex with more than one active site. Or you can say the area to be hydrolyzed could be between the same amino acids, because basically hydrolysis is going to break down the peptide bonds, because that is what it's going to do, because proteases do that, is break down peptide bonds. So you could have said quite a few things, but basically if you understood, because it says why is it possible for a protease to act on different types of protein? So basically active site and substrates have complementary shapes. Or you could have said substrates similar shapes, so fit the active site. So active site and protein have complementary shape. I've only written 1 point, of course, but there were many points for this. Then comes the C part of the question. Proteases known as keratinases vary in the extent to which they can hydrolyze keratin. Feathers are not easily degraded, broken down because keratin is a very stable protein. Suggests features of keratin structure that contribute to its stability. Now, there's something which we've got to understand is their syllabus point is the fibrous protein. Keratin is a fibrous protein, so it is insoluble, which means it does not dissolve, or you can say it does not dissolve easily. Then there's a high proportion of disulfide bonds, and disulfide bonds are covalent bonds, so very strong bonds. Then there's a high proportion of hydrogen bonds between the polypeptides. Then there is a high proportion of many hydrophobic interactions. Then there is a very close packing of the polypeptide molecules. Close packing. Do not say triple helix, that won't be allowed. There's a very tight or a close packing of the polypeptide molecules. So all these factors, or you could have also said is another point which is also mentioned, is arranged so the peptide bonds are not accessible. Parts of the molecules needing to bind to active site are not accessible because many amino acids are small R groups. So the fact that it is strong, there has to be something in the bonding or in the structure of it which makes it strong and gives it that strength. So you had to be talking about that. So any 3 points from that: insoluble, fibrous protein. This was given in the question. High proportion of disulfide bonds, and disulfide bonds are covalent bonds because disulfide bonds would be amongst the amino acids with a sulfur R group. High proportion of hydrogen bonds, high proportion of hydrophobic interactions, then close packing of the polypeptides. Now understand that is that they asked you a question is that it's not keratin is a very stable protein, and then it says suggest features that contribute to its stability. So here comes the sort of test of your knowledge, that would you know that what did we study which is very strong, and that was collagen. Collagen is in the syllabus, and collagen is a fibrous protein. It's insoluble. So even if you just wrote those 2 points, that is fibrous and it's insoluble, will get you your 2 out of 3 marks. Keratinases are used to degrade the large quantities of waste feathers from chickens and turkeys that are processed in the food industry. The products of feather degradation can be used in animal feed. Scientists investigated whether 3 different keratinases, K12, K12, A22, and P3, were suitable as industrial enzymes. These enzymes were extracted from 3 different soil bacteria. The effect of temperature and pH on the activity of each keratinase were investigated. The results are shown in figure 2.1 and figure 2.2. So we have K12, which is, let me give it another color. This is K12. So this would be K12. Please understand how we do these questions. Then this is A22. So A22 ये है. A22 is this. And then we have P3. So P3 is this one. So now let's look at them very carefully, look at their optimum temperatures. The optimum temperatures are different, but this is this optimum temperature. This one has this optimum temperature, and this one has this optimum temperature. So they're between 45, 50, 55. Different optimum temperatures, different peaks. Then you can say their range. The yellow one works between, green one works between here and here. So between 20 and 70. The yellow one works from here to here, and the red one works from 30 280. So the range is variable. So you have a look at it very carefully, think about it, process this information. And then we have there pH. So we have the pH on the x-axis. And let's look at it now here again. So we have K12. Let's look at K12. K12 works like this. Then we have A22, which is what the x's. And then you have the P3. So let's look at the P3 here. Now you see their optimum pH. So P3 has an optimum pH of 6, and this has an optimum pH of 7. This has an optimum pH of 8. So all this information needs to be read, needs to be analyzed, and then we go on to the question. To degrade feather waste from industry, it is an advantage to use keratinases that show at least 60% relative activity in conditions where temperature and pH can vary widely. Table 2.1 shows for each keratinase the working range of temperature and pH where at least 60% relative activity is obtained. Figure 2.1 and 2.2 to complete table 2.1 and use the completed table to name the keratinase that was the widest range of temperatures and name the keratinase that has the widest range of pH. So pH range, now we can see the pH range is 6.8 to 8.4. And then here we have the temperature range is 36 to 57, and that's it. So you have to finish this with 4 marks. And then it says keratinase with the widest range of temperature naturally was P3, and with the widest range of pH was A22. So that got you your 4 marks. Then it says some detergents contain proteases to remove stains from clothes. Like for instance, you drop some milk on it, it contains proteins. You drop some gravy on it or a meat broth on it, you dropped a piece of meat on it, some proteins in it. Some detergents contain proteases to remove stains from clothes. These enzymes have a high relative activity. These enzymes have a high relative activity in alkaline conditions. The scientists reported that K12 and A22 could be suitable for use in the detergent industry. With reference to figure 2.1 and 2.2, discuss the advantage and disadvantage of using K12 and A22 in the detergent industry. Now you have to go back onto the table and have a look at it. So the reasons you've got to understand is that if you look at this table, if you look at these two graphs, uh, you look at the two graphs and you have to look at the two graphs, and then you have to look at this table 2.1, which is here. So table 2.1, you can see A2 and K12. A2 and K12 are over a very wide range of temperatures. They remove stains at a wider range of temperature, that is 20, wide range of temperature. So their temperature range with at least 60% activity was quite large. And then low activity, poor stain removal. Disadvantage will be because you see it is poor stain removal at 30 and 70 or 80 for A22. So advantage and disadvantage. So you have to give me the advantage and disadvantage. So A2 advantage was greatest activity, most effective at pH 7.5 to 8. That's an advantage. Your disadvantage is that using figure 2.3 to show a decrease in activity at higher pH values. So from the graph and from the table, you had to pick up all the material, the advantage and the disadvantage. For the advantage for Temperature, advantage from figure 2.1 for the disadvantage for it, then advantage from figure 2.2 and disadvantage from figure 2.2. So you had to be really picking up things from here. You had to be picking up points from here. Advantage from figure 2.1. This is figure 2.1. Then advantage from figure 2.2. So 2.1 and figure 2.2. You had to be picking up figures from here. So you need to look at this graph carefully, look at the figures. Now I'm giving you the red are the advantages and the green are the disadvantages. So figure 2.1, A22 and K12 active at wide range of temperature. This was an advantage. Disadvantage was low activity. This is a disadvantage. The green ones are the disadvantage. Low activity at 30 degrees Celsius and at 80 degrees Celsius for A22. Then, then the for another advantage and disadvantage for the pH. So the advantage in figure 2.2 is A22 greatest activity at pH 7.5 and 8. And the disadvantage was from figure 2.2, you could find that it shows a decrease in activity at higher pH values. So you had to pick out the advantages and the disadvantages from the different graphs, and you had to go through the graphs again and again to really understand the question and answer this part of the question. The liver receives blood from the hepatic artery and from the hepatic portal vein. The hepatic portal vein transports blood from the digestive system. Hepatocytes are the main cell type of the liver. They have a wide range of functions, including the synthesis of triglycerides and plasma proteins, detoxifying waste, and energy storage. The hepatic artery branches from the main artery that transport blood from the heart. Name the main artery that transport blood to the hepatic artery. This would be the aorta. So you have to spell it correctly to get the mark for this. Then we come to the next B part of the question. Blood arriving at the liver enters specialized blood vessels known as sinusoids. Figure 3.1 is a diagram of part of a sinusoid and surrounding hepatocytes. A second type of cell found in the liver, a Kupffer cell, is also shown. Kupffer cells are phagocytic cells of the immune system. Large gaps between endothelial cells, Kupffer cells, sinusoid, and hepatocyte. Let's look at the question now. Suggest one advantage of having large gaps between the endothelial cells of the sinusoid as shown in figure 3.1. Now, why would they have gaps? There has to be some sort of a communication. So higher rate of exchange of substances. Substances do not have to cross a cell layer into and out of the endothelial cell. First enter the endothelial cell, then out of the endothelial cell. That's going to be very, very troublesome. So large molecules slash you can say plasma proteins can pass through, and Kupffer cells can pass through into the tissue fluid because they are phagocytic cells. So they can pass through. Phagocytic cells can move out of capillaries. So higher rate of movement of substances, large molecules can pass through, Kupffer cells can pass through. Part 2, in addition to removing bacteria present in the blood inside the sinusoid, Kupffer cells are also able to remove old or damaged red blood cells. Describe the mode of action of a Kupffer cell in removing and breaking down a damaged red blood cell. So what is it going to be? Endocytosis. Red blood cell engulfed. Forms phagocytic vacuole. Lysosomes fuse with the red blood cell, or you can say with phagosome. Lysosomes contain hydrolytic enzymes. Digestion to produce the heme and the globin, or the amino acids for the globin. Then, so You had a lot of points and you could have come up with any of these. Basically, it was endocytosis, red blood cell engulfed, forms phagocytic vacuole, lysosome fuse, hydrolytic enzymes released, digestion to produce heme and globin. So endocytosis, RBC engulfed, forms phagocytic vacuole, lysosome fused with phagosome, hydrolytic enzymes released, digestion to produce heme and globin. Then figure 3.2 is a transmission electron micrograph of part of a hepatocyte showing some cell structures. The peroxisome shown in figure 3.2 is a spherical organelle bound by a single membrane. It carries out a variety of enzyme catalyzed metabolic reactions, including detoxification. Some of these reactions require oxygen. So peroxisome, glycogen granules, liquid droplets. Describe the evidence visible in figure 3.2 apart from the presence of peroxisomes that indicate some of the functions of a hepatocyte. Add labels in figure 3.2 to identify the location of any cell structures if not already labeled that are part of your evidence. So smooth endoplasmic reticulum for lipid synthesis, rough endoplasmic reticulum for protein synthesis, mitochondria for many mitochondria for aerobic respiration. So you could have labeled these, or you could have labeled them, or you could have gone without the labeling as well. Then presence of glycogen granules as energy or a glucose store. Then presence of the lipid droplets again as an energy store. So you could have labeled any of these. There's the RER and the mitochondria. So it says able to identify if not already and they're part of your evidence. So the RER is for protein synthesis. So all these points were available to you. For a 3-mark question, we had something like 5 mark scheme points. So any three of these would have been fine. Then coming to part 2, the mitochondria in figure 3.2 are larger than the peroxisome. State one other difference visible in figure 3.2 between a peroxisome and a mitochondrion. Mitochondria is bound by a double membrane, and then peroxisomes, or you can say mitochondria has cristae and peroxisomes has no cristae. Then the peroxisomes are spherical shape and the mitochondria are different shapes. They're slightly sausage shaped, or you could say circular or spherical. Mitochondria has double membrane, has cristae, peroximum subferrical shape, and mitochondria different shapes. Then coming to part 3 of this question doesn't seem to end. Some of the enzymes used within mitochondria can be synthesized by the organelle. Peroxisomes cannot synthesize any of the enzymes that they contain. Suggest why the mitochondria can synthesize enzyme but a peroxisome. Well, naturally, mitochondria has DNA, has the genetic material, or you can say has DNA, and then it has the 70S ribosomes, which has the site for protein synthesis. So has the enzymes for transcription and translation. Also, it can produce ATP for enzyme synthesis. As you know, protein synthesis is a very important process for which energy is needed. And ATP is a universal energy currency. So mitochondria have DNA, mitochondria have ribosomes, mitochondria have the enzymes for transcription and translation, and mitochondria can produce ATP. So all the points, DNA, ribosomes. Please do not say ATS ribosome. That would be, you lose the marks. You can just say has ribosomes, and that would have been good if you're not clear about the 70S and the ATS. But writing ATS will give you, you'll lose a mark for that. Then coming to part 4 of the question, which finishes this question at last. Long question. One of the enzymes present in peroxisomes is catalase. This enzyme catalyzes the breakdown of hydrogen peroxide to harmless products. Suggest why it's useful for the cell for this reaction to take place within the peroxisomes. Now, information given to you. Now, peroxisome is membrane bound, so rest of the cell is protected from the hydrogen peroxide. So the peroxisome acts as a compartment or a specific area where an efficient breakdown of peroxide is going to take place. High concentration of enzyme in one location provides the optimum conditions for other peroxisome reactions. And then production of oxygen is a byproduct, so useful for peroxisome reactions which require oxygen. which require oxygen because the mitochondria can function and aerobic respiration can take place. Also, it isolates the enzymes from the rest of the cell. Like, in your house, you have your kitchen, so all the smell and all the cooking and all the byproducts of cooking, like all the waste, if you peel the onions and you peel the potatoes, all remain in the kitchen, but they don't spread in the rest of the house. So you have compartmentalized the cooking procedure in the kitchen. So just like that, the peroxisome is a compartment where you have isolated it from the rest of the cell, rest of the cytoplasm of the cell, where there'll be lots of other reactions taking place as well. But the peroxisome has localized it, has compartmentalized it, has kept it separate from the rest of the cytoplasm of the cell. So peroxisome acts as a compartment because it's a membrane bound, so rest of the cell is protected from the hydrogen peroxide, and high concentration of enzyme in one location, you can say, provides an optimum conditions for the other peroxisome reactions. Then production of oxygen is a byproduct, so useful for aerobic respiration. So any of these points, and you have to give me only 2 points to get your 2 out of 2. So this is a very easy question. I'm sure everybody could have figured this out, but be careful when you read the question, be careful when you write the answers. Thank you very much.